Great. Okay, in that case, I'll make a start. Okay, hello and welcome to this meeting of the Rotary Club of Leicester. It's the 15th of March. Good to see you all today, dressed very smartly and very nice to see. So, do we have any guests with us today? I can see Pam's folks. Welcome, Pam, as always.
Right. Thank you, and greetings from President Sarita and the Rotary Club of Leicester Novus. And you're all invited to our meeting this Thursday. I think you probably already had details of that.
Great, thank you. Could you just remind us of the time?
7:30 Thursday evening, Rotarian George Ritchie, a flying Rotarian. Should be very interesting.
Okay, thank you.
The incoming world president of the Flying Fellowship.
Sorry, say that again, Rodney.
George Ritchie is also the world president-elect of the International Fellowship of Flying Rotarians, and he gives a good speech.
Oh, great, great, thank you. Okay, do we have any other guests? I believe you have a guest with you, Gordon.
Yes, yes, my wife Janet is here with me.
I'm not wearing a jacket and tie, so you're just getting my head.
Okay, great to have you with us.
Thank you.
And I can't see any of the guests. No, okay. Any other greetings from other clubs I've missed? Okay, none, great. So as I said before, we're going to skip remarks and secretary's notices and committees, and we'll catch up with those next week. At this point, I'm very pleased to introduce our speaker, Dr. Julian Tang, consultant virologist at the Leicester Royal Infirmary. Dr. Tang is consultant virologist, honorary associate professor at the clinical microbiology clinical microbiology at the University Hospitals of Leicester NHS Trust. Dr. Tang trained in medicine and zoology at Cambridge before completing a zoology PhD in biological fluid dynamics in Aberdeen. Later the same year, he moved to Hong Kong after the SARS 2003 outbreaks. He later moved to Singapore in 2008 as a consultant virologist, arriving there just in time for the 2009 A1H1N1 influenza pandemic. He returned to the UK in 2014, settling in Leicester, UK, where he has been running the diagnostic virology laboratory and advising on the clinical management and infection control of viral infections, particularly respiratory viruses like influenza and SARS-CoV-2 that is causing the current COVID-19 pandemic. So very pleased to have such an eminent speaker with us today. And thank you, Julian. I believe you are here with us. So if you just want to say a few words, I think your telephone may be muted. Oh, I have muted it. Okay, so I can ask you to unmute that. Yeah, is that okay? Yes, excellent. Okay, it's working. So take it away. Thank you.
Okay. Okay.
You just need to turn off the sound on your laptop so we don't get any feedback.
Okay.
Okay.
It's not working very well.
Yeah, just
Better?
I think so, yeah.
I'll turn off the microphone.
That's perfect.
Yeah.
Thank you.
So thanks for inviting me to talk today. I've got a few slides covering pandemics, SARS-CoV-2 origins, transmission, infection control, vaccines and variants. And what I'll do for this audience, I'll start with the pandemic overview, so you know what pandemics are, and looking at the history of pandemics. So if you can get the first slide up, please, Pandit. So I can't see any slides on my end. I'm not sure what you can see from your end. Yeah, so the first picture is actually a picture of the 1918 flu pandemic, the Spanish flu. And you can see that even back then, all these people who were at that time were wearing masks as protection against the virus. If you go to the next slide, this is a nice kind of roadmap of all the different pandemics we've been looking at across the world within times where we can actually detect these pandemics, going back to the Justinian plague, the Black Death, smallpox, the more recent Great Plagues in London and Europe, and then even more recently yellow fever, cholera, and then HIV. And then at the bottom you can see MERS, SARS, swine flu. And the size of the kind of baubles are really the impact of those different pandemics on the global populations at that time. So you can see the biggest baubles, if you like, are HIV/AIDS, Spanish flu, bubonic plague. Look at the numbers of deaths caused by these infections. Now, of course, these are estimates. We didn't have PCR back then. We couldn't identify every death as related to these bubonic plagues. So these are really from historical reports, which are based on clinical and epidemiological assessments. If you go to the next slide, and this just demarcates and ranks them in a more kind of clear way. So the bubonic plague, Yersinia pestis, really has caused the most deaths in history, followed by smallpox, some way behind actually, and the Spanish flu, similar kind of level of deaths, and then plague of Justinian, which is of an unknown source at the moment. They think it may be Yersinia pestis as well, and HIV/AIDS, and then going back a bit further, Asian flu, Russian flu, Hong Kong flu. So it's kind of a mix of bacteria and viruses that cause these pandemics, and the pandemic is really loosely defined as an infection that has global impact. Now, exactly how you define global impact varies with the ages, and certainly with international travel, any potential infection outbreak has the potential to cause a global impact, if you like. Next slide, please. So just a background for those who are not medical, respiratory viruses, we have seasonal respiratory viruses every year. They normally peak during the flu season, the autumn-winter months. But there are some viruses that persist all year round, like the common cold viruses, the other coronaviruses, rhinovirus. They can persist all year round. The adenoviruses can persist all year round, at a lower level for sure. But during the winter, autumn months, they do actually peak in those seasons. And those of you who have grandchildren or young children of your own, if you remember that, a lot of them get colds and snuffles for the first couple of years of life. And that's really the human susceptibility to these new viruses, which are actually new to them. They're not new to mum and dad, who've been around for 20, 30 years. But to young children, they're essentially, all viruses are new, and they have no immunity once maternal antibodies have disappeared. So they're susceptible to all these viruses, and that's why the first few years of life can be very tiresome for mum and dad, bringing children back and forth to A&E or to their GP with bronchiolitis, croup, pneumonia, etc., whilst these children acquire immunity to these virus infections. The viruses cause the most respiratory virus infections globally, and also cause the most diarrhea-related illnesses globally as well. But after time, the children start to develop immunity, and they have fewer and fewer problems with these seasonal viruses. And that's partly one reason why the primary school vaccination program for influenza, which Leicester was one of the pilot sites for, has rolled out in the UK and has been elsewhere, in the US particularly, but also I think places like Finland and Austria, to try and reduce that that peak of influenza-like illness in the children, and also then trying to protect the adults who may be vulnerable to more severe disease in the household as well. Next slide, please. So this is what we do in the lab. So those of you who haven't been following the news very closely, we actually do swab and do PCR testing routinely every season. In fact, we run it every week for the whole year, looking for this kind of combination of respiratory seasonal viruses. You can see influenza A, B at top left there, parainfluenza, RSV, adenovirus, entero, and coronaviruses that I've mentioned earlier, as well as some other less well-known types like metapneumovirus, bocavirus, etc. And the PCR can be quantifiable, so we can actually see what the viral load is in the swab that you take from your own or someone else's mouth or nose. And that viral load can give us an idea of basically how much virus is there, how spreadable or transmissible this might be, and also how that might correlate to clinical severity of disease as well. And these numbers are not small. We're looking at 10 to the 10 viruses, which is like 10 billion viruses per swab, or as low as 100 to 1,000 viruses per swab, depending on the stage of illness you actually take the swab at. Next slide, please. So this is the culprit, coronavirus. The corona means crown. These are envelope viruses with crown-like spikes of glycoproteins on the surface that the virus uses to bind to host cells. that can allow entry and replication in those cells. And the receptor for this virus is something called ACE2, which is actually an enzyme used to control blood pressure. So anywhere where the cells are secreting this enzyme, expressing this enzyme, the virus can enter those cells. And a couple of coronaviruses use the same receptor, including SARS-1 that we saw in 2003, and also another common cold virus called NL63. MERS coronavirus and the other coronaviruses, 229E, OC43, and Hong Kong U1 use slightly different receptors and have different receptor distributions to some extent. And you can see on the bottom right, that's what the virus looks like in culture when you try and grow the virus. In the middle, the coronavirus looks like that under the electron microscope. That's the middle panel, if you want to point to it. And then the top right and bottom left are really just kind of cartoons showing a kind of stylized version of the virus and including some of the proteins you see. If you look at the bottom left image, the S protein at the top right, 1 o'clock position is where all the vaccines that you're getting are targeted. And that's the main binding protein to enter the host cell. So if you imagine the antibody binding to that protein, then that protein cannot bind a host cell. And there'll be an antibody for all of those proteins on the surface. So, there's multiple antibodies to multiple glycoproteins that stop the virus getting into a cell. If the virus can't get into the cell, it can't replicate. If it can't replicate, then it just sits around outside the cell, waiting to be gobbled up by macrophages, the white cells in your immune system. So that's why the vaccines are good. And the vaccines prevent infection and disease rather than you then having to fight the infection and disease with stronger and stronger drugs going forward. So prevention is really the key to preventing virus outbreaks and pandemics, rather than bacteria, which have lots of antibiotics but relatively fewer vaccines per se, where you can actually treat the bacteria. Antivirals don't kill the virus; they just prevent further viral replication. So if you've got 1 million viruses on Monday and they give you Tamiflu on Monday, if you don't clear that virus, you've still got 1 million viruses on Tuesday. You just haven't made any more virus. So your host immune system has to still remove that 1 million viruses that can still cause infection and disease in different cells in your body. So that's the drawback of antivirals, because they don't actually kill the virus. They just prevent further replication. Next slide, please. So where did this SARS-CoV-2 come from? Well, the recent WHO investigation into Wuhan and China, looking at the source of this virus, they declared that they didn't find any of the SARS-CoV-2 virus in that wet market in the samples they took way back in November 2019, January 2020. So although we suspected this wet market of harboring this virus, their samples at that time, taking their report at face value, obviously, because we can't do anything else, found none of the SARS-CoV virus in the food market, in the food stuff at that time. Next slide, please. So what they looked at is human cases in 2019 and found that in the humans they could find the virus, but just not in the food stuff in the wet market. So the human cases, as you see here, they were grumbling along for a while in December, early January, and then when they sent the CDC team, the Wuhan CDC sent a team in, and they found lots more cases when they started looking more actively and using a specific PCR test. So they had a PCR test way back in November, December that they were using to diagnose human cases at that time. Although we never heard about the environmental sampling that was going on, that they've now told us actually was negative for the virus. So where the humans got the virus now is one of the big debates going on within that WHO special investigation team. Next slide, please. So this is the wet market in its full glory. So you have, and I've been to wet markets in Hong Kong and Singapore, mainly Hong Kong, actually Singapore don't have any anymore, but you can buy all sorts of live animals there. And basically, we bought a chicken, and the chicken's running around in a cage, and they take out the chicken. If you choose that one, they cut off the head, let it bleed into a can, and then they have a defeathering machine that defeathers it, and they joint it, and by the time you come back 20 minutes later, it's already bagged up for you. And that's because the culture there is they like fresh food, so the wild animals do share cages in close proximity with each other, and they can transmit any zoonotic viruses between each other, which they would not normally do in the wild. And that's the main risk of the wet markets. We can have mixing of hosts carrying different viruses that normally don't mix. These animals don't normally mix, they avoid each other. But if you put them together, it's got snakes, bats, monkeys, or rodents, they can mix these viruses in close proximity, and that's where you get the problems arising. And you can see on the bottom left picture, you can't read the Chinese, but the images tell you've got peacocks, dogs, small mammals, and other types of birds for sale, including some, it looks like an ostrich at the top there. And of course the bats, and then the top right corner with the red ring, those are snakes in that store there. And the snakes are thought to be an intermediate host of this virus, as you'll see from the next slide. So we think that, bats carry the virus, snakes eat the bats, then the humans eat the snakes and bats, and then they get the infection from handling the raw meat from the snakes and the bats. And we know this happens because during HIV investigations, they found that the African bushmeat could actually harbor Ebola and HIV, chimpanzee HIV, and then humans get it when they prepare the meat, the bush meat, and get the blood of the animal into their skin via microabrasions. So a lot of this handling of the raw meat is the main risk, not the cooked meat, but the raw meat, the preparation step. Next, please. And of course, there's another, if you sequence all these viruses and you draw different trees from with viruses from different animals, you find that intermediate host, and the pangolin that you can see there's one of them. The pangolins used for Chinese medicine. They use the scales and the meat for medicinal purposes. And it is a very heavily trafficked illegal item, which they're trying to clamp down on. But a lot of this illegal animal trade has been moving viruses around the world. Certainly with West Nile virus in the US, there were rare birds being imported into the US from elsewhere that were carrying viruses. And even the monkeypox outbreak in the US back in 2002, they imported illegal African Gambian rats as a wild exotic pet into the US and spread monkeypox amongst the children who touched this big furry rat then as well. So lots of illegal movements of animals that could be carrying different viruses around the world. And you can detect this kind of relationship with viral sequencing and phylogenetic analysis, as you can see here. It's just like a family tree of viruses from different animals, and I also draw these trees as well for analysis. Next slide, please. So how does the virus transmit? So if you can just play this video, a lot of us think the virus transmits just by talking. So unmasked people talking, exchanging breath. So if I can smell your garlic breath or smell your alcohol breath or smell the curry you had for lunch, then I'm inhaling some of the air that could be carrying viruses as well. And this short range distance over about 1 meter, the corridor conversation or over a coffee table or just, at your desk could well transmit air laden with virus between you. You can see that's me on the left and my postdoc on the right. This was shot in Singapore when I had $1 million funding for this mirror imaging system. And you can see the air moving between us as we're talking from the nose and mouth. And certainly, if you've had this encounter with your colleagues coming back from lunch, you can smell what they had for lunch. You're inhaling some of the air they're breathing, and that could be carrying some virus that you're inhaling. And this happens every day between millions of people talking. And we think this is how the virus is mostly transmitted now, not by touch, not so much by large airborne volume routes, but really this face-to-face conversational contact. And that's why the mask works so well if you are talking to a stranger you don't know outside your household bubble. Next, please. Yeah. So this is just a graphic of this, and there's this controversy about airborne droplet nuclei, droplets. In fact, anything you can inhale is an aerosol. So whether you've got a 100-kilogram, 6'5" basketball player exerting himself heavily on a treadmill in a gym, or a young petite lady watching TV at home, if you can inhale the droplets, different sizes, because of different lung volumes and capacities, different inhalation forces, you can inhale the virus in those droplets or aerosols, whatever you want to call them. And if you can't, they drop on the gravity, land on surfaces, and you can touch them, then they touch your face and get infected that way. But in fact, there's no evidence for that either. There's no sequencing-related evidence for that particular thermite transmission route. So most of it we think is due to people talking over short distances, where we call it the garlic breath distance. So amongst the aerosol network I have, international network that we have that working with WHO, we call it the garlic breath distance. That if I can smell what you have for lunch, I'm at risk of inhaling your virus as well. Next slide, please. So the other thing that we found is that this virus transmits very efficiently pre-symptomatically, and for even up to 3 to 5 days before you get symptoms. And this makes it very hard to control. If you only test people who are symptomatic, by the time they become symptomatic, if at all, they've already had like 3 to 5 days of transmission potential before that. So you're always chasing the virus. The virus is always kind of spread before you've actually identified the index case, which is why these different variants going from door to door to pick up the South African variant or the Brazilian variant, if they've been infected and they've been out to the supermarket, for essential shopping, etc., the virus may have spread already by the time you identify them. So you're always chasing the virus. And this is why the UK has not been very good at controlling this virus, because they're always reacting, because they focus on symptomatic testing only. Especially at the beginning of the pandemic, they actually dropped community testing, focused on hospitalized symptomatic patients. And we know that only 1/3 of these infections, 1/3 of these cases are actually symptomatic, whereas 2/3 may be asymptomatic and spreading in the community. And that's why track and trace failed so miserably, because you can't track and trace when you've got 50, 60,000 cases a day. Like, if track and trace works really well in Hong Kong, Singapore, Taiwan, this type of Southeast Asian culture, where you're going to get, maybe less than 100 cases a day appearing, even at the peak of their pandemic. So I've been keeping in touch with my Singapore, Hong Kong colleagues, where, they're worried if they get 50 cases a day. And for a population that's maybe, 1/9 the size of UK, that translates to maybe just, 500 cases a day, rather than 50,000 that you get in the UK. And this is because the UK and the European countries left it too long to clamp down on this virus. And there's some data on this that I'll show you in a minute. So if you have a virus that transmits pre-symptomatically very efficiently, and the peak viral load we think is actually pre-symptomatic, then it's going to be very hard to control. Next slide, please. So this is a nice model study from back in 2004, post-SARS-CoV-1, smallpox, pandemic flu, and looking at HIV as well. And what you see is that on the far right, you see HIV. That's because most of the cases are transmitted through HIV before they get symptomatic. And you never catch this virus because you don't know who's infected. And those people who are infected don't know they're infected, and they carry on having sexual liaison with multiple people because they think they're okay. Unless you'll practice safe sex, which a lot of them don't do. In the middle, you've got influenza, which is kind of half-half. Influenza, we think, transmits for about a day or two before you get symptomatic. But the thing is, about half the people with flu don't get symptoms at all. So you've got the pre-symptomatic miss, plus you've got the asymptomatic miss as well. So you can never really control flu. And that's why it's kind of stuck in the middle there. It's not as bad as HIV, because you've got some vaccines, you've got some antivirals, and you can catch some of the symptomatics, but you can't catch all of them. And then on the far right, what you've got are the successful eliminations. So SARS-CoV-1 has disappeared, partly because SARS-CoV-1 is nearly always symptomatic. And when they present into hospital, they become more, the viral load increased when they're in hospital. That's why you've got so many healthcare worker infections. But if you eradicate the symptomatics, there are very few asymptomatics to worry about. Smallpox is the same. Nearly 100% of cases of smallpox were actually symptomatic. So if you identify them, quarantine the contacts, isolate the infected, and then you suppress the epidemic that way, and this is before PCR, okay, then you can actually effectively control that virus, because everyone who's infected is symptomatic. So you can just isolate them and quarantine the contacts, and you can control the virus very well that way. And measles is the same, chickenpox is the same, because they're although they're very infectious, they're almost virtually 100% symptomatic. And there's some pre-symptomatic transmission, but if you can identify all the ones who are symptomatic or have contact with those, you can control the virus eventually. Next slide, please. Now, what about masks? How well do masks work? So this is a nice little diagram that one of my followers drew and sent to me. So basically, if I'm wearing a mask, I contain my virus if I'm infected. If I'm not infected and I wear a mask, it protects me from the virus that comes towards me from you, for example. So if you're both wearing a mask, you can reduce that emission rate by about 4-fold, but you can also reduce the incoming exposure rate by about 4-fold as well. So you can reduce the overall exchange of virus between the two of you by 16 to 24-fold. And that's the benefit of universal masking. If only one person masks, then and I'm the infected one, I contain most of my virus in my mask, so you should be more safe. But if you both mask, that protection is more than doubled. And these are based on lab studies that, unfortunately, at the beginning of the pandemic, the UK and other countries said, masks don't work, we don't have evidence. Well, in fact, there was evidence, but they didn't know about it, or they didn't think it was evidence, because some people say lab studies are not good enough. Well, in fact, lab studies actually work quite well, because in the population, it's very hard to do an RCT, randomized controlled trial, because people don't keep the mask on properly, so you don't really see the benefit of the mask, and then you think they don't work. In fact, actually, they do work very well. You can look at all the Southeast Asian countries that have been using masks since the beginning of the pandemic and since SARS-CoV-1 in 2003, and they've got a few hundred cases at most with hardly any deaths compared to the Western European countries that have had thousands of deaths and tens of thousands of cases. Next slide, please. So this is one of the nice studies. This was done by the UK, HSE, Health and Safety Executive, back in 2008. So that's before pandemic flu, 2009, and before COVID-19, 2019. So there's very good evidence, and this is a very nice study done in a very rigorous manner, showing the benefit and the reduction of transmission with different types of masks. So you can see at the bottom right, there's an example here. The FFP3 mask, bottom right, is virtually 100% protection. Surgical mask at the far end is less. Okay, it's a lot less because it's a looser mask, but it's still reducing 4 to 5 times what you get otherwise. So a mask is better than no mask. Universal masking is better than just single masking, masking on one side or the other. And when I say a 6-fold reduction, it means that if 1,000 viruses are coming towards you, 6-fold reduction reduces that to 140. Okay, so you can reduce that virus load significantly, even just wearing a mask, just you on one side, for example. Next slide, please. Now, this is one of my colleagues, Don Milton, in the US, Maryland, University of Maryland, who showed that if you actually wear a mask, you contain the virus as well. You reduce that virus emission by about 3 to 4-fold. So if you can reduce the emission by 4-fold, if you're infected and you wear a mask, and you are wearing a mask to protect yourself from the incoming virus by 6-fold, the effect is multiplicative. So it's 4 times 6 is 24-fold reduction. So it goes from 1,000 down to 40 viruses, which is much better than getting a face full of 1,000 viruses coming in. Okay, but you've got to wear the mask properly. So you're not over your mouth and nose, but not under your chin or off one ear. Next slide, please. And children can wear masks. There are multiple examples of children in Southeast Asia that wear masks perfectly well, and they put up with it perfectly well because the culture there since SARS 2003, these affected countries had SARS 2003, they're aware of the risk. With a 10% mortality, they prefer to wear a mask than not. Next slide, please. Okay, it's all about vaccine. So looking at the makeup of the Rotary Club, I'm guessing that most of you probably had at least 1 dose of vaccine by now. So all of these vaccines probably work to some extent, even against the normal variant. So I just had the AstraZeneca last week. I got a bit of fever, a bit of chills, just like I do with the flu vaccine every year. But I was okay, I just took some paracetamol and it's fine. Most of the vaccines work against the UK variant and the original wild-type variant. There's some reduction against the South African variants, quite significant actually in some cases in the lab study, but in real life, the reduction is not that bad because you have T-cell responses and other cross-reacting antibodies that protect you. So the Pfizer-BioNTech vaccine is very good, Moderna is very good, the Johnson & Johnson is very good. It's just come out. The Novavax is very good. When I say very good, it's like more than 2/3 protection, it's like over 60% protection. The Russian Sputnik 5, I'm not seeing any data against variants just yet, or the Chinese Sinovac, Sinopharm, I'm not seeing any variants just yet, but that data will come out, and it may not be too bad. So what types of vaccines do we have? Well, here you can see the first one's protein-based. This is like the flu vaccine. It takes a bit of the virus, one of the virus proteins, amplifies them, purifies them, puts it into a vial, injects it into you. That's Novavax and the GSK Sanofi, and they work quite well. But they take a long time to produce because you have to grow the virus or produce the virus and then purify that one protein. The adenovirus vector vaccines, like the Oxford AstraZeneca vaccine, you need to insert a coding gene into that adenovirus, which is quick, and then you have to amplify that protein vector in the adenovirus vector to a large scale. So the adenovirus vector needs to be grown as well. So that takes a bit longer, but not much longer. And then the mRNA vaccines at the bottom are the fastest because you don't need to have a vector. It's basically just an mRNA code that you can modify very easily, very quickly, implanted in a liquid nanoparticle. But they've had the most frequent adverse effects actually compared to the other vaccines so far. So about 10 per million population have had some more severe adverse effects from Pfizer, BioNTech, and Moderna mRNA vaccines. but they've been rolled out first. So we may see more adverse effects with the Oxford AstraZeneca vaccine and some of the others going forward. And then normally local site injection, pain, tenderness, swelling, or a generalized fever, systemic fever, chills, headache, fatigue, which is what I had for a couple of days, but I resolved that on paracetamol. Next slide, please. So these are the vaccines that the UK has ordered. The vaccine types are shown on the right. We've just gone through those. So apart from the Russian Sputnik 5, UK has ordered the Pfizer, BioNTech, Moderna, AstraZeneca, obviously, Johnson & Johnson, the Novavax, and the Valneva, not the Chinese one, or the Sanofi GSK, because that actually didn't work very well. Then I think the next slide has a bit more detail on this. Okay, so this tells you about the prime boost strategy. So basically you have 2 doses. Why do you have 2 doses? Well, the first dose is to prime your system to that vaccine antigen, so the S protein normally, and natural infection does the same thing. So if you have the natural infection, that does the same thing, primes your system. And then the second dose is really just a booster to boost that response that you had initially. Now, we know that some people don't respond to the first dose. So the second dose becomes like their priming dose. And also, the results from the vaccine trials show B and T cell responses, which are good. But usually post-vaccine, we don't test you. So if there are 50 of you online now, the chances are that one or two of you may have failed the vaccine, at least the first dose. So if, say, 90% of you respond to the first dose and 90% respond to the second dose, then overall you get a 99% response to the vaccine. That works well with measles MMR vaccine. With this vaccine, we're looking at more like a overall, perhaps say, 60 to 90% response in the first dose, and if you don't respond, hopefully the second dose will catch you up with that. And in fact, there is some good data showing first dose responses can be quite good, and the first dose does most of the work for most vaccines anyway. Next slide, please. So this is actually the real list of what we've got so far. So you can see that I think GSK Sanofi have discontinued their efforts because it didn't work very well. But the rest of them UK's ordered, and you can see that UK's ordered 400 million doses of vaccine, which is enough to cover 200 million people, which is 3 times the UK population. Even if some people don't respond, there won't be that many. So hopefully we can give some of those vaccines to other countries that do need them. So at the moment we've got Pfizer, we've got Oxford AstraZeneca. I think Novavax is being approved. Janssen is a single-dose vaccine. That's also an adenovirus vector vaccine. That's being approved soon. So there's more than enough vaccine to go around in the UK, certainly. Next slide, please. And this is the kind of average cost in developed countries. I know COVAX has negotiated some free vaccines for other countries, but you can see Pfizer is the most expensive. Well, Moderna is the most expensive, followed by Pfizer, followed by the Sputnik 5, which UK has not ordered, and then the Oxford AstraZeneca, which is cheap. It's the cheapest because it's being produced and given out at cost by AstraZeneca. Next slide, please. So those of you who are more mathematically inclined, this is how they do the calculation. There's 1 minus relative risk. So if you know the numbers have had the vaccine and got COVID, and the ones that didn't have the vaccine got COVID, you can do a 1 minus RR relative risk and get those wonderful figures of 94% efficacy, depending on the data they present. Next slide, please. So the thing is, what is the end point of the vaccination? Is it to stop death? Well, most of the vaccines will stop death. Is it to stop severe infection, like ICU level infection disease severity? Most of the vaccines will do that as well. Is it to stop symptomatic infection? So PCR confirmed symptomatic infection, the efficacy starts to drop then because some of them still allow breakthrough infection and symptoms, but they're milder than if you didn't have the vaccine. Is it to prevent transmission? Well, it depends what you mean by that. So if you get the vaccine on Monday and you get exposed to the virus on Tuesday, you'll get infected because the vaccine doesn't work for at least 2 weeks. Okay, the vaccine produces antibodies that rise over time, but at a certain level of antibody, the infection can still be successful if the antibody levels are too low. Over time, antibodies will rise, and then they can fight the infection at different levels of exposure. So you might get infected on Tuesday and shed the virus until the antibody rises enough to neutralize that virus, in which case you might have spread it to some other people. No vaccine is going to work immediately. Okay, so you can get an instantaneous response. So if you get exposed while the vaccine is still taking, you can still get infected and transmit the virus to other people. And that can be what's happening in some parts of the population. Next slide, please. So what about variants? Well, if the S protein is the main target for the vaccine, and that S protein that looks like this normally, with the antibody that binds to it like this, induced by the vaccine, if the S protein changes shape, for example, the antibodies can't bind as well. Okay, they can still probably bind enough to reduce severe disease and death, but you may still get some symptomatic infection, disease that may allow virus to spread, like if you're coughing a lot or if you're still talking to lots of people. So that kind of change in the shape between the S protein might allow the antibodies to bind less well, reduce efficacy, effectiveness, and then let the virus spread a bit further before it gets neutralized. And this is the UK variant, it's the 501Y6970del deletion variant in the S protein. Next slide, please. Yeah, and then there's a South African variant that has 3 mutations of the S protein, the 501-484K and the 417N. The 484K is the main mutation that we think allows vaccine escape. So it changes the shape of the S protein sufficiently that antibodies don't bind as well. And the different vaccines have a different loss of efficacy to the South African variants. But certainly Moderna, Pfizer and AstraZeneca are now redesigning their vaccine to allow a third booster shot in autumn to protect against the South African variant. Next slide, please. This is just how the South African variant spreads. So the way you can estimate transmissibility is the predominance of that virus in the genetic sequencing that you do for the viral population as a whole. But this has some caveats. So for example, the South African variant was spreading in a very young population that has multiple contacts compared to the more elderly population. The UK variant had a lot of investigations about how it spread, but don't forget, during winter, when people are more indoors and pre-Christmas shopping, people are mixing more anyway, so that enhanced transmission may be just purely a seasonal thing that may not actually be due to the virus most of the time. So doing these calculations are quite tricky, and you need to be careful about over-interpreting the data. Next slide, please. So why has the UK done so badly? Well, I think most of the problem is because the UK and other European countries and the USA didn't really react much to the virus when it first presented in China, December, January. And it wasn't until it really hit Italy, and the UK had already had it by then in late January, early February, that you suddenly saw the rise of cases really explosively across that population, because basically there's no precautions, no interventions being taken. And UK had, the Cheltenham horse race back in March that really seeded the virus, let it spread. And once you've got a baseline of like 10,000 people infected, and each one can produce 2 to 3 other cases, that rise of cases will be very rapid and much too quickly to stop by track and trace, although it wasn't existent back then. Hence the first national lockdown. And the lockdown will work very well, because if you stop everybody from mixing, however it's transmitted, you will stop transmission of that virus. And if you keep them locked down for long enough, that virus can't go anywhere, and the numbers will gradually start to fall, which is what we saw in the March to June lockdown last year. Next slide, please. So I'm going to show you some data from Southeast Asia, because I know the people there and I've seen their response post-SARS 2003. You can just see the response. But basically, as soon as this appeared, all the maths went on. The track and trace teams just kicked into action, because they had this before 2003, MERS 2015. So all that infrastructure was in place. The personnel were in place. The systems, internet, mobile phone apps were very quickly brought online. So they were kind of already ready for this. Okay. So the UK had to learn this from scratch. So what they're good at in the UK is that, vaccine trials are good. They've been doing for years. Sequencing they've been doing for years. Antiviral drug clinical trials have been doing for years. But what they haven't got is that track and trace system to track the virus on home ground in a rapidly spreading situation. Next slide, please. So the first thing they could have done is put masks on. Now, if you masked everybody back in January, February, okay, I mean, some models done in the US and the UK that show that if you had this and other interventions, even a week earlier, they could have saved 20,000 lives, because if you delay for a week, so 10,000 becomes 30,000 within 7 days. And of that 30,000, 5 to 10% of them might end up in hospital, of which 1 to 2% may die. You have that kind of mushroom effect of spreading virus with hospitalizations and mortality arising from that. So with a pandemic situation, you must really go in hard at the beginning and stop the numbers from rising, because if you don't do that, you see what happens. Next slide, please. And you can see here the European studies, countries on the top, where the numbers have been rising since the beginning of the pandemic. Italy, US, Spain, Germany, UK, Canada, etc. They're all in the top part of this graph. Okay, whereas you see that countries that really reacted much earlier, who'd had this experience, Hong Kong, Taiwan, Singapore, South Korea to some extent, they'd already known what to do, and they put those measures in place without any mathematical modelling. You didn't need that. Everything went in place. In fact, they were asking for masks and suppressing numbers from day one. New Zealand should be there as well. New Zealand visited Southeast Asian countries and learned from them, and they took the decision to actually lock down very early. And you can see New Zealand's probably down at the bottom with Taiwan and Hong Kong. Next slide, please. So this is a slide I always put at the end. So is it going to disappear? No, I don't think so. Is it going to remain and become sporadic, like MERS and avian flu? Probably not. I think what's going to happen is that this virus becomes seasonal. You might need seasonal vaccine boosters like for flu every year to protect the vulnerable population. And this will continue until the end of time. I think this virus will become seasonal like the other coronaviruses, but with that extra additional mortality on top of that, which might need the vaccine. We don't need vaccine to control the common cold coronaviruses, but you may need the vaccine to actually reduce the mortality and morbidity of this COVID-19 virus going forwards, like with the seasonal flu. And the final slide, I think. Yeah, so this is not the only virus. There's another virus that people are monitoring in southern China amongst the swine population. It's called this Eurasian avian-like flu illness, and it has pandemic potential. So we're watching this, I think it's called a G4 virus at the moment. And that's it. Hope I haven't gone too much over time, but I'll try and answer any questions you have.
Thank you, very informative, Julian. So at this point, I'm just going to open it up for questions. If you have a question, I see Rodney's hand is up already. Please put your hand up and unmute yourself. Thank you. Go ahead, Rodney.
Questions in two parts. We've seen that the manufacturers, some of the vaccines recommended a 3-week for the second dose, and it's actually being done as 12 weeks. I'm interested in your comment of that. And also, there's been a suggestion that people would have a second vaccine. I'd be interested to know what you say about that. And
okay, so I think, sure. So I think both of those are really no problem. In fact, we do some mixing and matching of vaccines inadvertently anyway. So the first dose does most of the work. In fact, the vaccine response may take a while to appear, maybe more than 3 to 4 weeks. Some of the Oxford-AstraZeneca clinical trial data shows that, in fact, the vaccine response from the first dose is really quite effective up to like 100 days. That's like 3 months. Okay, so there is data showing that the first dose does a lot of the work, and in fact, the time of the second dose could be delayed with even more effect later on. So I have no problem with that. For a second vaccine, a second dose of a different vaccine, I also think that's probably okay. So if you're going to have a second dose of the vaccine later on, like 6 to 12 weeks later, okay, there shouldn't be any major problem with that. It's just the booster, as long as it's boosting the same target antigen as the first dose, then you get that boosting effect. So if you have like AstraZeneca first, Pfizer-BioNTech second, or Moderna second, or Pfizer first and AstraZeneca second, I don't think that's a major problem either. But at the moment, they're trying to avoid that, but they're doing some clinical trials to look at that. In fact, we know this happens with, say, the pneumococcal vaccines. So my son actually had 3 different pneumococcal vaccines. He had the Prevnar 7, GSK 10, Prevnar 13, all within the first year of life, and he's perfectly fine because they're targeting the same antigen, and the immune response will just catch on to each vaccine booster that he gets.
Thank you.
Thank you. Tony.
Yes, thank you. Could I just ask you about herd immunity, which I don't think you really covered, about the only thing you didn't cover, I think, but we were told some time ago that once the population has had 60, 70% vaccinations, that herd immunity would kick in, and we don't need 100% vaccination. Is that correct?
Yeah, probably not. You don't need, so the herd immunity is calculated by a simple equation. It's 1 minus 1 over R0. Okay, so if R0 is 2, you just need 50% herd immunity. 1 minus 1 over 2 is 50%. Okay. This virus, you think, probably because of the variants, is transmitting a bit more quickly. So it could be like 1, it could be like 2 to 3 effectively in the population. So 1 minus 1 over 3 is 67%. 1 minus 1 over 4 is 75%. So it's not an exact calculation because the herd immunity, basically what you do is you immunize everybody. So even if there's some kind of breakthrough infection and transmission going on, that transmission bounces off a wall of immunity given by the vaccines. But if you've got variants like the Brazilian variant or the South African variant that breaks through that vaccine barrier, like maybe, say, 2 out of 3 times, okay, that 67% herd immunity may not be enough to protect against all those different variants as well. That's why they've now increased that to like 70 to 80% just to be safe.
So if we didn't have any variants, we'd only need 50, 60% coverage.
Exactly.
But the variants have extended that.
Yeah, if you have the same Wuhan original virus that had back in January last year, probably only need 60% coverage. So the current vaccines could give you that by the end of the year, for example.
Thank you.
We've got time for one more. Patrick. Please unmute yourself. There we go.
Okay, yeah. Thank you very much indeed for that. Going back to the origins and so on, I've been involved for more than 20 years internationally with cave science, and 10 years ago at least, we started getting groups exploring the deep caves in southern China and other parts of the region, getting unknown variety of pneumonia, and so at least 2 or 3 deaths from it. And we started putting out the advice that people going into horseshoe bat caves should always mask themselves and so on. So that's possibly one of the areas where it came from. I mean, certainly endemic, and there is a nasty virus endemic amongst the horseshoe bat populations of southern China and Vietnam and surrounding areas.
Yeah, so bats are a reservoir for many viruses. We think Ebola, Nipah virus, coronaviruses, other human zoonotic viruses may be a reservoir in bats. And the Chinese team have actually been looking at bats in caves for many years, since SARS 2003, 17 years, picking up all these different coronaviruses and other viruses as well. So they're still, this is ongoing surveillance that they've been doing since 2003. But they said that a lot of the caves around Wuhan didn't show this virus either. So that's why they've now got this story about imported from other countries through food products, et cetera, et cetera. So that still is under investigation. But certainly we know that bats are a reservoir for many different viruses that can be potentially hazardous to human health.
I should say that I'm chairman of a small nature reserve in Devon, which has the largest population of horseshoe bats in Britain. And so we're a little bit jumpy about these things.
Well, I hope you've had your rabies prophylactic vaccine.
I don't think we have rabies now. We'll get that on the. We've got a council trustees meeting on Saturday, and I'll get that on the agenda.
So you must have rabies surveillance for those bat populations. Certainly, some of the Scotland bat populations do carry Lyssa virus, which is the rabies equivalent for bats. And there is a recommendation to have rabies vaccine if you're going to handling those types of animals.
Well, of course, under English law, you have to have a very special license to literally to handle any bats, from a baby little ones up all the way through. So I guess that our members, that we've only got, I think, 2 members who are actually licensed to physically capture or handle bats. But it's a thing we're more to warn about. I think just guest visitors going in to look at the caves and so on are probably going to be okay. But and we don't have access to the caves at all anyway during the high risk parts of the year, because they're then breeding. But thanks for that. Yes, I raised a new thing to consider on Friday and Saturday.
Okay, thank you. I will now hand over to Richard Power to give vote of thanks.
Right, so we're a year on since this all started, and being the only medically qualified actively working doctor in the club, it's been my pleasure as a barber, surgeon, bonesmith to talk to you on a weekly basis, and so it's a refreshing change, I'm sure, for everybody to actually have a proper doctor talk to you. And I think it's fair to say Julian is a proper doctor. As I was reading the Sunday Times yesterday, up popped his name being quoted. And when I phoned him last week for a chat, he just got off the phone to BBC Brazil. So we have certainly heard from greatness today. But no, in all seriousness, I think you've given us a very full, comprehensive overview of everything COVID. Which even I understood as an orthopedic surgeon, so that's high praise indeed.
That's good.
And all I can say is we look forward to hearing you again this time next year when we finished year 2 of the pandemic and we've had the Armenian variant and the Leicester Golf Club has been ploughed up as socially distanced municipal allotments. But anyway, we do that, honestly, Julian, it's been a fantastic talk, and I'd ask my fellow club members to thank you in the usual way.
Well, thank you. You've been a very good audience and some very useful questions there.
Yes, and you're more than welcome to come and visit again as our guest once we start meeting again over at the Mercure Grand Hotel on Granby Street.
Yes.
We'll send you an invite.
Okay,
wonderful. So it's been very interesting. It's been great to mark our 1-year anniversary since our last meeting. And we are trying our best to get us back to physical lunch meetings. Oh, sorry, please everybody go ahead and unmute yourselves. So in 2 to 3 weeks, just so everybody knows, we're looking at dates for when we can return, obviously subject to government restrictions and things like that. So we are working hard in the background, trying to understand how that's going to look. So that's that. I guess we don't need a COVID-19 update from Richard today. Just in case, yeah. So we do normally have an update from Richard on that, but I believe Julian
can give it for a change.
So thank you for that. So next meeting we have Roger Graves, retail manager at HSBC Grantham, talking to us about how to avoid banking scams. So looking forward to that. And attendance today, yes, I think it'll be one to watch. So attendance today has been 26. It's been great to have you all on. See you all next week. Stay safe, stay well, and goodbye. Take care.
Bye, everybody.
Bye.
Bye.
Bye, everybody.